Okay, good morning, students. Today's lesson is very, very short, maybe about 20 minutes. We're going to look at objective number 11. Okay, objective number 11. And what is this in this? What is stated here? Explain the principal operation of dipstick. So we're going to look at dipstick and biosensor for measuring glucose only. So glucose. We're not going to measure other substances like urea, hormone, ketone, yeah, drugs. Is only glucose present in the blood and urine with reference to glucose oxidase and peroxidase. Okay, so these two enzymes are very, very important. Yeah, very, very important. Glucose oxidase and peroxidase. Now, when we talk about blood, so normally this is for diabetic, those who suffer from diabetes. And urine, this is for those who have kidney problem. Okay, if there's glucose, monitoring glucose in your blood, normally this is for diabetic patients, and monitoring glucose in the urine, this is normally for those people with kidney problems. Okay, so again, I want to highlight glucose oxidase and peroxidase. Right, let's go back. Let's go for. So what are dipstick and biosensor? So dipstick and biosensor, a lot of you are very familiar already with dipstick, yeah. Particularly nowadays, we've been using the art kit, yeah, once every week. So the art kit is more or less like a dipstick, yeah. The only difference is that you're not dipping, because you are basically you have this sample here. You have the to show the band there, and then you put your sample here, and then you're gonna drop your nasal proteins, in case if it contains viral proteins, onto this sample, and then you will see that there will be two bands positive, and there's only one band if it's a negative. Okay, so this is more or less like a dipstick, because there's also a art kit that works like a dipstick, yeah, where you immerse this, yeah, you immerse this into the tube. You have a tube here, and there's a solution here, and this is why it's called dipstick. Okay, but principally, in principle, the dipstick and the art kit, they work in the same way. Yeah, they work in the same way. Okay, so what are these dipstick and biosensors? These are biological molecule. They use biological molecule to detect and measure the concentration of certain substances. It usually consists of a membrane to which a layer of enzyme or antibodies or receptor molecules is attached. So normally if it's antibody, it's a monoclonal antibodies, one specific type of antibodies. Now we have already you have already learned this, the meaning of monoclonal antibodies, how it is made, yeah, mixed with cancer cell to from hybridoma and so on and so on. Okay, so that's the monoclonal antibodies. So normally it's monoclonal. In the case of the art kit, it's using monoclonal antibody looking for viral proteins. Okay, so if it's regardless if it's of course COVID-19, they have different strains nowadays. There is used to be Delta and then Omicron, but basically all of these different types of strain, they contain one specific protein, yeah, one very, very specific protein, and that's why it can be tested. You know, the COVID-19 art kit can be tested, be it if it's Delta or Omicron. it can detect, because both Omicron and Delta have one specific type of protein. Apart from the other proteins, of course, they have this one common protein. Okay, let's look at the glucose, urine glucose test. Okay, two equations, very important. So this is the reason why it's called dipstick, yeah, because you are dipping it into a beaker which contains your urine. So this is talking about urine. Likewise, the art kit can also be used, can also be dipped into a very small container, yeah, very, very small container. Okay, it works in the same way. All right, now you have to memorize these two equations. These two equations are very, very important. It's stated in the learning objectives. Okay, glucose oxidase and then peroxidase. Okay, how does this work? This works by oxidizing glucose, yeah, into gluconic acid and hydrogen peroxide. So this is oxidation. It's an oxidation process. Okay, so if there is glucose in your urine, more glucose in the urine will be oxidized by the glucose oxidase to form more hydrogen peroxide. Okay, to form more hydrogen peroxide. And then the hydrogen peroxide will oxidize a chemical indicator. So this is a chemical indicator, which is also present on the dipstick. Okay, into this. So it will be colored. So the color of this chromogen color depends on the amount of glucose in the urine. Okay, so let's so all these two enzymes, glucose oxidase, peroxidase, and the chemical indicator, they are already attached to this dipstick. Okay, already attached. Okay. So these are enzymes which are normally immobilized. Yeah? Immobilized means more stable from the effects of pH, from the effects of temperature. Yeah. So when it's being transported from one place to another, it's not easily denatured. Yeah, because it's already immobilized. So whatever your results, you will compare it to a chart. So this is a chart to determine the concentration of glucose. Yeah. So it will be different from each individual. So don't forget the two equations. Yeah, very, very important. Two equation, two oxidation process. Okay, glucose oxidized to gluconic acid, hydrogen peroxide, and then the chemical indicator here oxidized to chromogen, which is colored. Okay. The chemical indicator has many different types. Remember, this is the chemical indicator. There are many different types of this chemical indicator. In this case here, it's colorless, and then it will be colored, yeah, depending on the amount of glucose. In other textbook, like this one here, the chemical indicator is blue. Yeah, this is the chemical indicator. Okay. So the same thing, yeah, two equation, the first equation and then the second equation. Yeah, glucose oxidase, peroxidase. Yeah, both are oxidation process. Okay, glucose is oxidized to gluconic acid and hydrogen peroxide, and then this chemical indicator is also oxidized to chromogen dye. Yeah, so the color will depend on the amount of glucose. If it remains blue, that means no glucose is present. If it remains colorless here, that means no glucose is present in your urine. That means you're good, you're healthy. Okay. All right, so these are the two equations that you must memorize. Okay. Now let's look at, we can read this. The dipstick has the enzyme glucose oxidase on its surface, which oxidizes glucose present in the urine and blood into hydrogen peroxide, gluconic acid. The hydrogen peroxide then oxidizes and indicates the chemical which changes color, the presence of peroxidase enzyme. Then you compare it to a chart. Okay, so memorize these two equations. These two equations, very, very important. And then we look at biosensor. So biosensor, this is normally used for blood. Yeah, biosensor is normally for blood, measuring what's in your blood. Okay, so you need to pierce a sharp needle on the tip of your fingertip, and the blood comes out, and then this is the special piece of paper that has the biological molecule. Okay, you can buy this, of course, from pharmacy, Guardian, okay, clinics, private clinics, okay, you for stocking, yeah, for stocking up. So this is the device. Yeah, the device itself, it doesn't have a biological molecule, but you need to buy this thing, yeah, this special piece of paper, yeah, which you can buy from pharmacy, Guardian, or clinic. Okay, so there are many different types. For glucose, that it will be specially for glucose. Yeah, for urea, it will be specially for urea, and so on. Okay, so so how does it work for the biosensor? Now, for the biosensor, there is only one reaction, and that is the enzyme is immobilized glucose oxidase. Yeah, make sure you know what it means by immobilize. Immobilize means stable here. Normally it means stable from the effects of temperature, pH. Yeah. So for the biosensor, there's only one chemical reaction, and that is the glucose oxidase. So the same, yeah, the reaction is the same. As you can see here, glucose plus O2 to form gluconic acid plus hydrogen peroxide. The enzyme is glucose oxidase. This is for biosensor. So for biosensor, the more glucose present, more glucose present means there will be a drop in the oxygen to make more hydrogen peroxide. Okay, so the higher the glucose in your blood, more oxygen will be required to make more hydrogen peroxide. And so the dipstick will measure the drop in this oxygen. It says here, an electrode measures the drop in oxygen concentration that is used to make hydrogen peroxide. The electrodes will create a electrical signal. Okay, so remember there's only one equation. Right, there is a, don't forget that there is a transducer as well present, yeah, in the biosensor. This is amplifying chemical reaction to electrical signal, or converting chemical reactions to electrical signal. Okay, like here, If we look at this one here, there's a selectively permeable membrane here. So selectively permeable membrane here will filter the unnecessary substances. So we will only allow this thing, and it will not allow the circle here. Just like when you are performing the art kit, yeah, the art kit, sometimes there's a cotton bud, yeah, there's a cotton here. Okay, now this cotton, if you insert into your nose, it will pick up the protein, viral protein, of course, mucus, and then of course sometimes they can be blood as well. Blood, red blood cells. And so the selectively permeable membrane will only allow perhaps only the viral protein and maybe the mucus, but not the red blood cell. Okay, so that is the purpose of this selectively permeable membrane. So the binding to an enzyme, the binding of this triangle shape to an enzyme or a monoclonal antibody will cause a chemical reaction, chemical reaction, and that chemical reaction is transformed to electrical signal by a transducer. There's a transducer in the biosensor. Okay, this is taken from another textbook. Okay, so only this will be allowed to enter. Okay, this one will not be allowed because they are perhaps filtered out. Okay, so there's a chemical reaction here, amplified by transducer, electrical signal, and then you will see a certain figure. Okay, so the biosensor has many purposes. You can do it to test urea in your blood, yeah, but of course you need to have that special piece of paper that you insert here. Okay, you can measure it for ketone, you can use it to measure drugs, metabolites, anything, for as long as you know you have that special piece of paper. This is an old picture to measure glucose. So this is the thing here that you buy from clinics, pharmacy, Guardian. This one is the one that contains the biological molecule. So this device itself, this is just a device, so it doesn't have the biological molecule in it. This is the one that has the biological molecule. Okay, advantages of using biosensors, they're very specific, yeah. The same thing with the dipstick. The dipstick is also very, very specific, yeah. It will only measure a particular substance. Rapid response, the dipstick is not rapid, yeah. The dipstick is slow. Dipstick is slow. In fact, if you notice with some of the low quality art kit, yeah, it takes about 10 to 15 minutes. Safe to use, accurate, can be very, very small, can be mass produced. Okay. Problems of biosensor, they are not very robust, meaning break easily. If it falls on the floor, it can break, yeah. They are not very stable because they're operated by battery, yeah? So that means you must have if the battery is weak, then it will not give accurate reading. So operated by battery, they are not sterilizable. Okay, so medical uses in your syllabus is only glucose, but it can also measure protein, ketones, metabolites, drugs, hormones, and many, many others. Okay, so that's all for today. We'll continue again tomorrow on tomorrow's lesson will be on stomata. Okay, that's all for today.